Hello friends. In today's video we will talk about collagen fiber. Uh, what is collagen fiber? It means introduction of collagen fiber, synthesis of collagen fiber, types and location of collagen fiber, and collagen fiber related disease. So this is a very important topic. Every medical topic is very important. So let's talk about introduction. Uh, so collagen fiber is most abundant, most it is most abundant protein in our body, and it consists of almost 25% of dry weight of total dry weight, 25% of dry weight, and uh, it is made up of peptide chain and three peptide chain by made up of three peptide chains. These peptide chains may be uh, same, these three peptide chains, or may be different to each other. So collagen fibers are different types of according to their location. Now let's talk about synthesis of collagen fiber. It is very important. See, collagen fiber synthesis is mainly occur in fibroblast and other cells of the body. So let's assume this is fibroblast. Then fibroblast have nucleus, and within nucleus, this is nucleus, chromosome present, and on the chromosome there is DNA present, and this is the gene synthesis for collagen fiber. So first it form the mRNA, then it go to the cytosol, and here this mRNA uh, bind with ribosomes, and after binding the ribosome, uh, protein synthesis will start, and first the sequence is formed. So when this mRNA bind with ribosome, translation occur, and first the segment which is formed in the cytosol, which is signal sequence. So when protein translation occur, first signal sequence form. This signal sequence is hydrophobic. Hydrophobic. So it easily cross the membrane. Let's see here. It is a organally present inside the cell, ribosome. So this endoplasmic reticulum, it is endoplasmic reticulum. So this signal sequence with ribosome came, and this signal sequence penetrate this endoplasmic reticulum and enter, but ribosome will bind here, not enter in the cell, uh, in endoplasmic reticulum. So there are many ribosomes come and bind, and protein synthesis and further translation occur. So therefore this ribosome is called rough endoplasmic reticulum, because the ribosomes are stick here, and this is now not smooth, this is rough endoplasmic reticulum. So when the signal sequence enter, and translation will occur continuously, then the full peptide chain will form. After entering in and endoplasmic reticulum, this signal sequence will degrade and become disappear, but all new synthesis protein sequence or chain will occur. This is known as pre-pro alpha peptide. This pre-pro alpha peptide, and it further go in the ribosome for modification. So let's see, here is two enzyme presents, uh, one and two enzyme, and in the sequence of pre-pro alpha peptide, every three every three amino acid each in each chain, every third amino acid is glycine. So what is the function of glycine? Glycine help the pre-pro alpha peptide in twisting. Because glycine is very small molecule, therefore when three alpha peptide bind to each other, the glycine help, because it is very small molecule. So with the help of glycine, the twisting occur very powerfully. So it is a function of glycine, and glycine is present on every third position in pre-pro alpha peptide chain. So when this pre-pro alpha peptide chain go further, and so this further go to the for modification, uh, and now here is two enzymes. First I want to tell you that before the glycine there is a amino acid is present, which is or uh, proline or lysine. Before glycine there is a amino acid, it is a proline or lysine. So here is a two enzyme is present. Number one is a propyl hydroxylase, and number two is Lysyl hydroxylase. Uh, and here is a molecule present, very popular molecule, it is vitamin C. So when this pre-pro alpha peptide go further for modification in the endoplasmic reticulum, here the two molecule propyl hydroxylate and lysyl hydroxylase is present. The function of these two molecules are uh, uh, hydroxylation. The function of these two molecules are hydroxylation. So here is glycine, here is glycine, here is glycine. Before the glycine, proline or lysine is present. So the either of these enzyme, if there is lysine, then propyl, uh, if this is lysine, then lysyl hydroxylase work, and hydro and hydroxylation reaction occur. This hydroxylation reaction make here OH molecule, and this OH molecule uh, work as hook molecule. When the pre-pro alpha peptide chain form the uh, collagen fiber, then this OH molecule function work as hook, so twisting will occur better firm. After and these two enzymes are work in the presence of vitamin C, and after it this pre-pro peptide go further in the endoplasmic reticulum, and here one enzyme is also present, which help in glycosylation. glycosylation at hydroxyl point. So when it comes here, then glycine molecule is here. Before glycine, valine or lysine is present, which is hydroxylated by these two enzymes. Now this is glycosylated. Here it glycosylate by the enzyme present here, and it is more efficiently twisted to each other. So after it, this molecule pre-pro alpha peptide is go out from the endoplasmic reticulum by the vesicles, secretory vesicles, and here is endoplasmic Golgi apparatus is present. In the Golgi apparatus, this pre-pro alpha peptide came, and here another enzyme is present which uh, further glycosylate this molecule. And now see here, this is glycine on every third position. Before glycine, proline or valine is present, this one, and it is a hydroxylase by the enzyme propyl hydroxylase or Lysyl hydroxylase. After it is glycosylated by this enzyme present in endoplasmic reticulum. Now it is further glycosylated in the Golgi apparatus and become more sticky to each other. So this is now become secreted by the Golgi apparatus by the secretory vesicles, this molecule, and go to the outside of the cell. See here, I make it long. So every third position is with glycine. Before glycine, this is proline or lysine. It is hydroxylase by the enzyme and function at hook, and glycosylate by the endoplasmic reticulum enzyme, and further glycosylate by the Golgi apparatus enzyme. And now and in the endoplasmic reticulum, the twisting process will also occur. See here, in the endoplasmic reticulum, this is endoplasmic reticulum. These molecules, alpha peptide, 3 alpha peptides bind with each other, but the side of these are very loose, not compact. After this process, this go to the Golgi apparatus, and so here is also triple helix, and here is also triple helix. It means 3 alpha peptide chain is present, but on the side they are very loose, not so much compact. So there is an other enzyme present from the extracellular environment. This enzyme is known as collagen peptidase. This collagen peptidase cut these loose ends. This loose end cut by this collagen peptidase also here, this cut. And when this collagen peptidase is cut, then only the tightly twisted alpha proteins are present here. Now here is another enzyme present which is called please see here first this what is this molecule. This molecule is called tropocollagen molecule. Now it is tropocollagen, and when this molecule is cut, this form tropocollagen, and here is an enzyme present known as lysyl oxidase. Lysyl oxidase is present here. What is the function of Lysyl oxidase? The Lysyl oxidase further oxified this chain on which point where is lysine present. So this is more sticky, and this tropocollagen are accumulate here like this. So when Lysyl oxidase oxidize these three chains at the point of lysine amino acid, then this tropocollagen will stick by this point with nearest tropocollagen. So tropocollagen are stick to each other by the help of Lysyl oxidase on the many points, and so this whole tropocollagen molecule is now called fibril, and many fibrils accumulate and form collagen. This is now collagen fiber. So this is the whole process of the synthesis of collagen fiber. Some process occur in the intracellular, while other process occur in extracellular environment, and the other type of collagen fiber is also present. I will discuss after some time. So let's say synthesis is occur, and types and location. So this type of collagen synthesis in which fibril are present, this is the type 1, type 2, type 3, type 5. But type 4 and type 7. Type 6 is not present in so much amount, so it is not so much important. These collagens are very important. So collagen type 1, type 2, type 3 and type 5 contain fibrils, and collagen type 4 and 7 don't contain fibril. How collagen type 4 and type 7 synthesize? See here. Here is the enzyme present, collagen peptidase. In synthesis of collagen type 4 or type 7, collagen peptide is not present. So when collagen peptide is not present, then this loose end of the alpha peptide chain will not cut by this enzyme. So another peptide which is synthesized in the intracellular environment, it bind on the end of this chain. So here is a structure form like meshwork network. So meshwork network here is form. So this type 4 and type 7, mainly type 4 collagen fiber is synthesized by this type. What means collagen peptide is not present, so the another triple alpha peptide chain or pre-pro collagen combined with another pre-pro collagen and meshwork network form, and this is collagen type 4. So collagen type 1, 2, 3 and 5 contain fibrils, while collagen type 4 contain meshwork network like this. So what is the location of these fibrils? Types, collagen types. First, collagen type 1. It is present in bone, as here is 1%. Collagen type 1 is present in bone, and collagen type 2, it is present in cartilage. And collagen type 3, it is present everywhere in body, it is free. Collagen type 3 is free and present everywhere in the body. And collagen type 5, it is present in embryonic stage. And collagen type 4, it present in glomerulus. And it also present in the inner ear. And collagen type 7, it present in between dermis and epidermis. So these are the collagen types and location. Now we will talk about the disease in collagen synthesis. The first disease in collagen synthesis will occur, and it is known as osteogenesis imperfecta. So first disease is osteogenesis imperfecta. How it's occur? See here, when protein synthesis occur on the every third molecule, every third amino acid on peptide chain, every third amino acid is glycine. So glycine is very small molecule. So the three alpha chains twisted each other easily by the help of glycine, because it is very small molecule. But the in osteogenesis imperfecta, the gene which synthesize the mRNA, which will become mutant. And on the space of glycine, there are any other amino acid which contain large molecule, which is large uh, form. So the twisting will not occur properly, or very loose twisting occur. So therefore collagen fiber not bind to each other properly, and bones will become weak. So it is the osteogenesis imperfecta. And osteogenesis imperfecta is very dangerous, because if it occur in intrauterine life, then the bone will become so much weak, and by the some pressure of the uterus, bones become rupture and intrauterine death occur. Uh, this disease is known as osteogenesis imperfecta. Osteogenesis imperfecta congenita. Osteogenesis imperfecta congenita, if intrauterine death occur. But if the child will born after because this process the bones become weak. So after birth, the baby will be very weak, and by the uh, from very small injury become bones become fracture. So this is known as osteogenesis imperfecta terda. And if it occur in uh, mature life, then bones become very weak, and commonly known as osteogenesis imperfecta. And these all three diseases are commonly known as brittle bone syndrome. brittle bone syndrome. Brittle bone syndrome is occur by the mutation of the gene which synthesize the amino acid which or peptide chain by the third position of glycine. But in this case the third position is not acquired by the glycine, it is acquired by any other amino acid which is large molecule. So twisting will not occur properly, and bone and other connective tissue become very weak. So osteogenesis imperfecta occur. And the second one disease is scurvy. How scurvy occur? See here, when pre-pro alpha peptide form, uh, then it further go in the endoplasmic reticulum for modification. Here is a propyl hydroxylase and lysyl hydroxylase is present, which hydroxylate the pre-pro alpha peptide at the point of lysine or proline is present. But these two enzymes are work in the presence of vitamin C. When vitamin C deficiency occur, or vitamin C is not present, then these two enzymes will not work. When these two enzymes will not work, then before the glycine hydroxylation will not occur. So and we know that hydroxylation is occur, and after hydroxylation there is hook like structure form. These are help in twisting the alpha peptide to each other. When it is not occur, then these hooks will not form, and when these hooks will not form, then twisting will not occur properly, and collagen fiber become very weak. So in this type capillary become rupture, or mainly it's seen in uh, gems. So here the blood leak out from the capillaries to the interstitial fluid, and it is also dangerous, and if it is prolonged, then it cause the death. So it is very important to care about it. And in this case the joints become very loose and very flexible. You can move in either direction of the joints. So this is the second disease caused by the collagen synthesis. And the third disease in this case, this is Ehlas danlas syndrome.